Hey everybody, welcome back. Today we're going to go into the first topic of virology. Viruses are acellular and incapable of self-replication in that it needs a host cell to assemble all of their components. And they encode any required processes not provided by the host cell in their DNA, which can either be DNA or their RNA. I'm sorry, I meant by their genome. So the genome can be either DNA or RNA. So the smallest human DNA virus is the parvovirus. Remember, that's really problematic because the parvovirus, especially parvovirus B19, can cross the placental barrier and affect the fetus. The largest is going to be the pox virus. And the smallest human RNA virus is going to be the picornavirus, and the largest is going to be the paramyxovirus. Virion are going to be your latent viruses. These are not going to be like the active virus. So they're going to be composed of a capsid like surrounding the nuclei, and it's made out of a capsomere protein. Besides protection, it has some host cell attachment properties. The genome and the capsid together combined makes the nucleocapsid, and these can be icosahedral or helical. So some of them have an envelope surrounding the capsid, and that is going to be made from the host membrane through budding. Peplomers or spikes are made out of glycoproteins, and these are your attachment molecules. They attach to the host receptor like ACE through like a variety of subunits, and these will determine the virus's host range and cellular tropism, so what the virus is going to bind to. They have the matrix proteins or M proteins, and those will mediate the attachment of the capsid with the envelope, and those are going to allow the transmembrane transmembrane transport of nutrients. So the NE and other accessory proteins are going to interfere with the host response in order to allow the virus to continue to either infect the existing cell or bud out and infect more cells. The packaged enzymes are going to facilitate the initial viral replication. So these are going to be the polymerases and the proteases. And inside the nucleocapsid, or in between the space between the capsid and envelope. And the structural proteins that are part of the virion are not part of the packaged enzyme, they're part of the actual genome. The non-structural proteins are not going to be within the virion, they're going to be encoded in the genome. So first, there's recognition of the viral genome, and then it binds, and then there's going to be packaging using ATP. The nuclease is going to be stimulated, and then the mature infectious virion is made. Okay, so the naked icosahedral is a polyhedral structure. As you can see right here, the icosahedral is like a 20-sided ring, not just a ring, it's a 3D structure, right? And they are not covered in an envelope, that's why it's called naked. These include the poliovirus, the adenovirus, and the hepatitis A. The naked helical are going to be capsomeres arranged in a hollow coil, and this is only the tobacco mosaic virus. The enveloped icosahedral is going to be covered by the glycoproteins, and then between the capsid and the membrane is something called a tegument. And then it's going to be the herpes virus, the yellow fever virus, and the rubella virus. The enveloped helical is going to be a bullet-shaped under microscopy, and these are going to be your rabies virus. along with the paramyxovirus, which includes the measles and the mumps, and the parainfluenza virus. The complex structures are going to be the special ones, and these are going to be your poxvirus and your bacteriophage. Okay, so the paramyxovirus are going to be non-segmented negative sense RNA, and okay, so I guess I should explain what's a negative sense and what's a positive sense. Positive sense RNA allows the host cell, once infected, to just produce the viral proteins. There's nothing needed, right? There's no translation needed. It can bind to the host ribosomes immediately and start cranking out viral proteins. A negative sense needs something, so it carries its own equipment. So it will carry the RNA-dependent RNA polymerase. So the RNA-dependent RNA polymerase is going to make the viral proteins, thanks to a peplimer with the F protein, and that's going to allow the virus to go into the host cell by inducing the fusion between the viral envelope and the cell membrane. There's usually some spikes with hemagglutinin protein or neuraminidase protein, so those are going to help to release the virus from the infected host cell. The capsid in the genome can be the same but different in the types and the composition of spikes, thanks to like conjugation, transduction, all of the ways that viruses evade the host immune system. The influenza virus is a segmented negative sense single-stranded RNA genome. Its peplimer is a hemagglutinin, and it also has neuraminidase, which split the sialic acid to be released. In doing so, it decreases the neutrophil response because the neutrophil needs to have the sialic acid intact in order to bind to the receptors. So in splitting the sialic acid, it also makes the mucus runny. That's why you have like a runny nose. And since it's a negative sense, it's going to carry its stuff. It's going to carry its RNA-dependent RNA polymerase. HIV is going to be your segmented RNA, and this is unique because it has two positive single-stranded RNA strands enveloped within a helical capsid. So the surface glycoprotein GP120 and GP41 will allow the virus to dock onto the helper T cell, and the GP41 is going to be your transmembrane protein, so it's going to allow this to be injected into the T helper cell. Since it's a positive stranded, it doesn't have to carry the polymerase, but it does carry reverse transcriptase because it's a RNA virus, and integrase and proteases. The coronavirus is going to be a long positive sense single-stranded RNA. It has the peplimers or spikes on the surface in a coronal pattern, and it's enveloped with a helical capsid, so very tricky to treat. The capsid prevents opsonization, and the peplimers will decrease the amount of immune response. So as you can see here, spike protein will allow the binding to host cells. Envelope protein E interacts with M to form the envelope. And variola is smallpox. It's a double-stranded DNA virus. It's completely enveloped in its complex structure. It has like a dumbbell on the inside. If you're looking at cut like sagittally, it would look like this. So here's the dumbbell, lateral body. You have to remember that it's enveloped, and within or in between the lateral body and the envelope is the soluble protein antigen. Okay, so an enveloped virus is environmentally labile, so it's susceptible to detergents, lipid, solvents, and dehydration. It's spread by droplets, secretion, and blood because it needs the liquid. It needs to be hydrated. So they enter the cell by fusion with the cytoplasmic membrane lipids, and it's released by budding. And it can spread from cell to cell thanks to syncytia. We'll go through the process of syncytia later. The naked viruses are environmentally stable. They're spread by fomites, contamination, droplets, human contacts. So this doesn't rely on the presence of liquids to remain infectious. And they can enter the cell through the receptor-mediated paths, and they exit the host cell by lysis. So this is lysogenic because of the release of budding, and this is going to exit by cell death. DNA viruses are 7 families. It can be the icosahedral, enveloped, double-stranded, non-enveloped, single-stranded or double-stranded, and complex. So the double-stranded linear DNA virus is the herpes virus. Double-stranded circular is going to be the human papilloma virus, and those are and then the partially double-stranded circular viruses are the adenovirus. Single-stranded linear virus is going to be the parvovirus, and all of these need to replicate in the host nucleus except for the pox virus because of its complex structure. It has an ability to partially replicate in the cytoplasm. All of these, since they have to enter the nucleus and they mess around with the cellular mechanism, it has some neoplastic potential. You got to remember the DNA virus are your happy viruses. So herpes, hepatodNA, adeno, parvo, pox, papilloma, and polyoma viruses. There are far more families in the RNA virus category, and they replicate in the host cytoplasm except for orthomyxoviruses. The retrovirus will replicate in both cytoplasm and DNA, and nucleus. The negative sense viruses are all enveloped, and they carry with them their RNA-dependent RNA polymerase. Only the RNA viruses can be borne by bugs because they're the smallest. Other non-cellular infectious agents, prions, viroids, satellite or defective viruses. So these need a co-infection because they need other viruses to stabilize it or code for other materials that they're missing. The adeno-associated viruses replicate only in the cells infected with adenovirus. And then the delta agent is going to be a naked strand of RNA, and that's only expressed in the presence of hepatitis B. First, the virus will come and attach to the target epithelial cell, and then it is going to penetrate the cell due to either endocytosis or their molecular needle. The viral nucleic acid are going to be released during uncoating, and that creates the mRNA from the viral genome and the copies of the viral genome itself. The macromolecular synthesis is going to occur, and then there's going to be assembly, and then there's going to be the release due to budding or cell lysis. The viral attachment proteins, or VAP, are going to be your peptimers or spikes, and that determines host tissue specificity. There's a huge mutation rate change in VAP, and that can change the host specificity or avoid the immune response. Those will bind to the CD4 and CXCR4 or CCR5 on the T helper cells and the macrophages for HIV. ICAM1 on the epithelial cells are going to be bound by the rhinovirus. And the sialic acid on the epithelial cells are going to be bound by the influenza virus. And all of this is going to allow the permissiveness of the cell to increase. Okay, so the enveloped viruses are pH dependent. They are going to fuse with the host membrane at neutral pH, which is unfortunately blood pH. at the acidic pH, they are endocytosed. So if they are in contact with, or they're trying to affect an unhealthy cell, the unhealthy cell is going to decrease their pH. The naked viruses will go through receptor-mediated endocytosis, and the viral pexins are going to just like directly inject itself into the cell. And then the capsid is going to be digested or opened, and then the nucleic acid is going to be released. So here's the viral pexin, the endocytosis of the naked cell. Okay, so macromolecular synthesis is one of the main goals of the virus. It needs mRNA, and it's going to make its regulatory proteins along with other proteins it needs to be a virion. There can be either early or late mRNA synthesis, and then the protein is going to be synthesized. So they make the amino acid, the fatty acids, which are a huge part of your phospholipid membrane, either carbohydrates, glycerol, or monosaccharides, and nucleobases in order to replicate. Okay, so that's it for now. Thank you for your attention, and good luck studying.